Now, the first thing we need to know is what is the difference between eukaryotic and a prokaryotic cell. Now, prokaryotic cell is a bacteria and eukaryotic cells are plants and animals. Now, if you see in this diagram, there is a eukaryotic cell and a prokaryotic cell. The first basic difference between the two is the presence of this structure called the nucleus. So, nucleus is present in a eukaryotic cell, but in a prokaryotic cell, the nucleus is absent. Apart from the nucleus, eukaryotic cell, you can see they have those organelles which have membrane around them. So all membrane-bound organelles are present in a eukaryote, example mitochondria, but these membrane-bound organelles are absent from a prokaryotic cell. The next difference is, since the nucleus is absent, in the eukaryotic cell, the DNA is enclosed in the nucleus, but in a prokaryotic cell, it lies naked in the cytoplasm as it has no nucleus. The eukaryotic cell are mostly multicellular, but the prokaryotic cell are mostly unicellular. The eukaryotic cell DNA is linear, but prokaryotic cell DNA is circular. Both eukaryotic and prokaryotic cell has ribosomes, but in a eukaryotic cell, the ribosomes are big. In a prokaryotic cell, they are small. Eukaryotic cells are multicellular big cells, whereas prokaryotic cells are small unicellular cells. Example is a plant cell and an animal cell of a eukaryote, and for a prokaryote, there's a bacterial cell. So I hope you're able to write these differences, because in the exam, they can ask you to list, any two differences or any two similarities between a eukaryotic cell and prokaryotic cell. So make sure you understand these points. Next is, let us look at in detail the structure of an animal cell. Now the diagram here shows you the animal cell, and you can see there's a round big nucleus. The jelly-like fluid that fills the cell is the cytoplasm. The outer layer that surrounds the cell is the cell membrane. And then in these structures, on them and freely, you have ribosomes. And then you have these pink structures called the mitochondria. Now, you should know these names and the functions of each, as these are found both in plants and an animal cell. So the first is the nucleus. It is the brain of the cell, because it controls all the activities. It controls what is happening inside the cell, and it also has your genetic information as it contains the DNA. So you should know all these three functions of the nucleus. Next is the ribosomes. Ribosomes are the site for protein synthesis. Now the cell needs proteins for its functioning, and enzymes are also protein. So ribosomes, they make proteins and the enzymes which are required by the cell. Next is the cytoplasm. The cytoplasm is like the jelly-like fluid which fills the entire cavity of the cell. It is a very important part because it has all the enzymes for the chemical reactions of the cell. So majority of the chemical reactions of the cell takes place in cytoplasm. Next is the cell membrane, as it's the outer layer. So as it's the outer layer, it is controlling what is going in and out of the cell. And mitochondria is very important. It is a powerhouse of the cell. Powerhouse means it provides the cell with the energy, as it is the site where aerobic respiration takes place. Okay, so I hope you know the function of each of these organelles. Okay, and you should be able to label them. If any cell is given to you, you should be able to spot where's the nucleus, where's the cell membrane, where's the mitochondria. Next is the plant cell. Now the plant cell has all these organelles which we have just discussed in the previous slide, except it has three extra organelles which are just found in a plant cell. This is a big, large permanent vacuole. These green structures called the chloroplast, and outside the cell membrane there's an extra layer called the cell wall. Now, the permanent vacuole, you should remember the function of each one of them. The permanent vacuole is filled with the cell sap, and it gives rigidity to the cell and makes the cell turgid. The cell wall is the outer layer, and it is made up of cellulose. You should remember the cell wall of the plant cell is cellulosic. It is a layer outside the cell membrane, and it supports the plant and maintains its shape. The chloroplast is the site of photosynthesis, because it contains a green pigment called chlorophyll, which absorbs sunlight and converts the light energy into chemical energy, making food. Okay, so you should know the difference between an animal cell and a plant cell. So this is a table that summarizes the difference in both plants and similarities. So in the exam, they can ask you to, give two similarities or two differences. So you should know both similarities and differences in both of these cells. So both are eukaryotic. The nucleus is present in both of them. Cell membrane is present in both of them. Mitochondria is also present in both of them. Ribosome is also present in both of them. And cytoplasm is also present in both of them. But cell wall, vacuole, and chloroplast is present only in the plant cells. So it is absent in an animal cell and it is present in in a plant cell. Okay, so this difference is very, very important. Next is the bacterial cell. You should know a typical prokaryotic cell, which is a bacterial cell. Make sure you can label it. So you can see the tail-like structure, which is the flagellum, which helps the bacteria to move. Then it has a cell wall and the cell membrane. But the cell wall is not made up of cellulose. You have to remember here, it is made up of peptidoglycan. Then outside the cell wall, you see a slimy layer, which is a capsule, and this capsule protects the bacteria. And you also see some hair-like structures outside this cell wall. These appendages helps the bacteria to reproduce. Okay. Then, since it has no nucleus, the DNA is circular and it is found naked in the cytoplasm, and it has some extra chromosomal material. You can see here they have plasmid. Now, plasmids are the extra chromosomal materials which are found in the bacteria. So you can see these extra stretches of DNA. These are plasmid, and they are in the form of small rings, and they give special properties to bacteria like antibiotic resistance. Okay, so I hope these all organelles, this labeling and their functions are clear to you. Okay, so now this is similarities and the differences between the bacteria, plant, and an animal cell. Bacteria is prokaryotic, plant and animals are eukaryotic. The nucleus is absent in bacteria but present in both plants and animal cell. Cell wall is present in bacteria but non-cellulosic. Remember, in a plant cell it is cellulosic, in animal cell it is completely absent. Cell membrane is present in all. In bacteria, ribosomes are smaller, but in plant and animal cell they're bigger. DNA is circular in bacteria but linear in plant and animal cell. The genetic material in bacteria is naked, found in the cytoplasm. In plant, it is in the nucleus inside the chromosome, and same in animals. The chloroplast are absent in the bacteria but they are present in the plant and animal cell, and the vacuoles are small in bacteria but in the plant and animal cell they are absent in the animal cell but present in the plant cell. Okay, so I hope the similarities between bacteria, plant, and animal cell and the differences, they are clear to you. Okay, so next, in your syllabus, there's a topic on order of magnitude. Now, what is order of magnitude? They'll give you the size of the two cells, and then they'll ask you what is the order of magnitude of the two cells. So what you need to do is, you need to divide the size of the two cells, okay? And that will give you an order of the magnitude. So for example, if one cell is, let's say, the 10 meter, and the other cell is, let's say, 100 meters, right? So you divide the two sides, so you will get the answer as 10. So it means that one cell is in the order of magnitude 10 than the other. Okay, so for that, it is very important the units are same. So in the exam, they can give you one in meters, another in nanometer, one is in micrometer, another in millimeter. So you should remember this conversion. So this conversion is simple. It comes as a chart: killing metal Milo, Mickey nano pictures. Okay, so killing metal Milo, Mickey nano pictures, it's kilometers, meters, millimeters, micrometers, nanometers, and picometers, and all are at the level of 10 to the power 3. So kilometers to meters times by 1,000, times by 1,000, times by 1,000. And when you go reverse, you need to divide by 1,000. So for example, if you need to convert the nanometer into meter, so you're going from here to here. So you will divide it by 10 to the power 9. Okay, so 1 nanometer is 1 times 10 power minus 9 meters. So you can remember this table, and this is a mnemonic to remember it. So if it is 10 nanometer to meter, so from nanometer to meter, what do? You divide it by 10 to the power 9. So that becomes 10 power minus 8 meters. And micrometers to meters, so micrometers to meters is 1, 2. 2 bumps, so it means divide by 10 to the power 6. So that gives the answer 10 power minus 5 meters. So it's very important. You know how you need to convert. Make sure both the units are same and then divide the two. Okay. Now, the next thing we need to do is, what are the specialized animal cells? Now, what are specialized animal cells? Now, specialized animal cells are those cells which have some extra features that allow them to perform the specific functions. Okay. Now, What you need to do for each of these cell type, you need to know what are the functions of each of these cell type, and depending on the functions, what structural characteristics they have. So for example, a nerve cell. What is the function of the nerve cell? To send electrical impulses around the body. Now this is the structure of the nerve cell. And in this nerve cell, what we have is that the nerve cells have dendroid. Now, what is this dendroid? These dendroids are the hair-like structures that receives the impulses. Then it has a long stalk called the axon. This axon transmits the nerve impulses, and at the end you have the synapse. Now, what is a synapse? They transmit the nerve impulses from one neuron to another. So the structure goes like this. It has a nucleus, hair-like structure, and a long stalk, and at the end it has the synapse. Next is the muscle cell. The function of the muscle cell is to contract and bring about the movement of different parts of the body, and for that they are made up of the special fibers which helps them to contract and relax. They contain special proteins that allow them to contract and relax. They have loads of mitochondria which provides them energy to contract, and they have special storage carbohydrates called glycogen which acts as a fuel for the muscles. Okay, so for both of them, you should know what is the function and what features do they have. Next is the sperm cell. Now, if you look at the sperm cell, it looks like this. At the top, there's a head which has an acrosome, and this acrosome contains hydrolytic enzyme which breaks the egg cell wall and penetrate inside the egg to fuse with the egg nucleus. The middle piece has mitochondria. The mitochondria provides the energy to swim. The nucleus contains the genetic information, and the flagella is a tail-like structure which helps it to swim to large distances. So I hope this function, and you should be able to label this, like what is an acrosome with its function. Okay, the flagella with its function. So you should know the different components of a cell along with their functions. Okay, so now as we have finished the specialized animal cell, in the same way we have the specialized plant cell. Now, what are the specialized plant cell? Again, these are those cells which have some extra features that allows them to perform special functions. Now, again, to write about these, you should first know their function. So root hair cells is a cell which act as an exchange surface and absorb water and minerals. Now, for the absorption of water and minerals, it needs a large surface area. So the root hairs are the extensions which provides a large surface area for the absorption of water and minerals. Then it has a large permanent vacuole which supports the cell and speeds the movement of water by osmosis. At times, it's not just the diffusion through which the water and minerals move. The active transport also happens. So for that, there's a mitochondria inside the root hair cell, which provides the energy for active transport. It has a cell wall, and it is just one cell thick so that the substances can easily diffuse and the diffusion pathway is reduced. On the other hand, there's a xylem cell. What is the function of the xylem cell? It transports water and minerals from roots to all parts of the plant. Now, since it has to travel water to large distances, it has to form a long continuous column. Now, the cells of the xylems are dead so that they can form a long continuous column which can transport water to large distance. And then inside, you can see the lignin deposit. The lignin deposit makes it waterproof and makes the xylem column strong and strengthen the xylem vessel. The phloem cell is again a transporting tissue, but its function is to transport food from the leaf to all parts of the plant. Now, the phloem has lost majority of the cell content so that it can have more room for the food. The cells at the periphery, so it is in the form of the sieve tube, and the cells which are at the end of the sieve tube, they are broken to form a sieve plate so that the food can easily percolate. Okay, and since the sieve tubes have lost majority of their cell contents, they are supported by a special cell called the companion cell. It supports the phloem cell and provides the cell whatever they need. Especially they are rich in mitochondria, which makes a lot of energy for the sieve plates and sieve tubes so that they can transport food. The last is the photosynthetic cell. The function of the photosynthetic cell is to prepare food by photosynthesis. So it has a large vacuole which helps in the movement of water by osmosis. It is having loads of chloroplast that helps it to trap light for photosynthesis. Also, these cells lie flat onto the leaf surface near to that so that they can easily absorb water, carbon dioxide for quick photosynthesis. Okay, so I hope these special plants and animal cells are clear to you. Now the question comes in, the cells we cannot see by the naked eye. So we take the help of the microscopes. So microscopes are the devices that use to see the cells which we cannot see by our naked eye. And there are two properties of the microscopes which are important and you should know the definition of them. First is the magnification. Magnification is the property of the microscope to enlarge the object, and resolution is the property of the microscope to distinguish between two closely placed objects. Now, magnification is calculated by the image size over an object size. Now, this is an important question. In the exam, they can give you an image size and they can ask you to find the object size if the magnification is given, or the image size and object size is given to you and ask you the magnification. Or to make it a little complicated, they can give you a cell and they can say that this diameter is 2 micrometer in reality, calculate the magnification. So in that case, what you'll do, you'll take your ruler, measure this actual length, that will be the image size. The object size, they have already given you 2 micrometer. Make sure you make the unit same by the chart that I have just discussed. I'll just put it again. And then you find the magnification by doing the image size over the object size. Okay. So the table that is given before helps you to convert the different units into a single unit. And remember, for the magnification, both image size and object size should be of the same unit because magnification is the ratio. Okay, so now there are two types of microscope. First, we had the light microscopes. These are the microscope which you see in your school laboratory. They are easy to handle, small and compact, does not require much expertise to handle, and they just have either a mirror at the bottom or a bulb, as it uses the beam of light to focus on the object. Whereas electron microscope, it is not easy to handle, it is big and non-portable, requires proper training, it uses beam of electron to focus on the object. Okay, now when we use a light microscope, we cannot see the greater subcellular details because the light microscope has a very low resolving and the magnifying power. The resolving power is 0.2 micrometer and magnifying power is just between 1000 to 1500. Whereas for electron microscope, it has a greater resolving power around 0.5 nanometer and the magnifying power is around 100,000 times. So it is the electron microscope that have enabled us to see a proper subcellular structures like mitochondria, ribosomes, and chloroplast. But for the light microscopes, you can view the live samples and you do not require special preparation of the samples. But for the electron microscopes, the samples have to be dead, and we require some special sample preparations. Light microscope, we can just see the color images. We can stain the sample and see the color images. Whereas electron microscope, it can form 2D and 3D, but the images are black and white. Okay, so I hope this difference between the light microscope and the electron microscope is crystal clear to you. Okay, so make sure you can write the similarities and the differences between the two. Okay, so now you should be able to know the difference between a plant cell and an animal cell, the bacterial cell, the light microscope, and the electron microscope. So now we have looked in what are the different structures found in the cell and what are the functions of each of the cell organelles. Now, in order for the cell to survive, the cell need materials. The cell need oxygen, the cell need glucose, the cell needs proteins, enzymes, and on the other hand, the waste products are also produced in the cell, so the cell needs to get rid of them as well. So there need to be the substances to move in and out of the cell, which happens through the cell membrane, but there are two processes that are involved in movement of substances in and out of the cell. So we categorize these two processes in terms of active transport and the passive transport. Now, in order to understand this active transport and passive transport, let's take an example of a hill. So there's a hill and one boy is standing with the ball at the top of the hill and one boy is standing with the ball down. Now, it is easier for the one at the top of the hill to send the ball down, and it will not take much of the energy to send the ball down. But on the other hand, if you send the ball up, you require more energy because it is going against the gravity. The same thing happens in terms of transport. Whenever the particles move from low to a higher concentration, which means the particle move against the concentration gradient, it requires energy, and this is known as active transport. And when the particles move along the concentration gradient, that is from a region of higher concentration to a region of lower concentration without the use of energy, then that is known as passive transport. Now, since the active transport requires energy, and we know it is the mitochondria that is responsible for making energies for the cell, so the cells which are involved in active transport have loads of mitochondria, and in passive transport, having numerous mitochondria is not a requirement. And the passive transport is of two types, diffusion and osmosis. Now, let us see what is diffusion and what is osmosis. Now, if you see this diagram here, this was an empty container of water and a drop of ink, which was purple in color, was dropped. This ink started to spread out, and it spread it out evenly until the particles of the ink are evenly distributed in the water. You can see this with this example. There are more particles onto the left side as compared to the right side. So the particles with time from the left side move towards the right side until the concentration of the particles is equal on both sides. This phenomenon is diffusion. Now, if it comes in the exam, how will you define diffusion? You will say it is the net movement of particles from an area of a higher concentration to an area of lower concentration. It is a passive process, so it does not require energy as the particle moves along the concentration gradient. Now, what are the factors that affects diffusion? Now, the diffusion is dependent on four important factors. One is the surface area. Second is the concentration gradient. Third is the diffusion distance. And fourth is the temperature. Now, greater the surface area, greater is the rate of diffusion of the particle, because if the surface area is more, the particle will get more room for movement. So all the exchange surfaces have a greater surface area, like the root cells has root hairs and intestine has villi. The greater the concentration gradient in the two regions, greater is the rate of diffusion. So all the exchange surfaces, they maintain the steepest concentration gradient. How? The root cells are close to the xylem, and villi has rich blood supply that maintains the concentration gradient. Now, diffusion distance has to be smaller for the diffusion to take place, because the particles, if they travel lesser distance, they will diffuse quickly. So all the exchange surfaces maintain a smaller diffusion distance by being one cell thick. So if you see any of the exchange surface, it will be one cell thick. And last is the temperature. Greater the temperature, greater is the rate of diffusion, as the particle will get more kinetic energy for movement. So to summarize, I can say that rate of diffusion is surface area times the concentration gradient divided by the diffusion distance. And you need to remember that, because if any questions on adaptation of diffusion comes in, you need to talk about all these four factors, how that particular surface is maintaining a higher surface area, greater concentration gradient, smaller diffusion distance, and temperature. Now, the diffusion in animals, there are two examples for diffusion in animals. In lungs or the alveoli, there's a diffusion of gases that takes place. And in small intestine, diffusion of digested food takes place. If you see this structure of the alveoli, the lungs have millions of alveoli, which increases the surface area. For the shorter diffusion distance, they are one cell thick. And for maintaining the concentration gradient, they have rich blood supply. On the other hand, the small intestine, the wall is folded to form finger-like projections called the villi. Villi are one cell thick and they have rich blood supply. So if this question comes in the exam, what are the adaptations? You need to talk about all these three factors: how the largest surface area is maintained, how the diffusion distance is maintained, and how the concentration gradient is maintained. Now, another example for diffusion in plants is diffusion of gases through the stomata and diffusions of water and minerals through the stem. So for stomata, it has a greater surface area because it has tiny pores called stomata. For thin diffusion distance, they are one cell thick. And for steep concentration gradient, photosynthetic cells are close to stomata. For the root hair cells, the root cell project to form the root hair cells. Root hair are one cell thick, and to maintain the concentration gradient, xylem is located close to the root hair cells. Okay, so I hope the examples of diffusion in plants and animals, along with the adaptation they have for maximum diffusion, is clear to you. Now let us look at another special case of diffusion called osmosis. Now, why osmosis is a special case? Because there are two special things in osmosis. First, it is the diffusion of only water molecules. Diffusion can be diffusion of any substance, but osmosis is the diffusion of just water molecule. And second, it is a semi-permeable membrane or partially permeable membrane is required. Now, what is a semi-permeable membrane? The membrane that allows only specific molecules to pass through, like water. Now, if you see this diagram here, where there is more water, the water is more in the left because there's no solute. On to the right, there are salt molecules, so it has less water. So water from the dilute solution, dilute means has more water, passes through this concentrated solution through this semi-permeable membrane until the water concentration is equal in both cases, and this is known as osmosis. So osmosis is the net movement of water particles from the region of high concentration of water particles to a region of low concentration of water particle across a semi-permeable membrane, or you can say it's the movement of water from a dilute solution to a concentrated through a semi-permeable membrane. So I hope this definition of osmosis and this concept that it is the movement just of water molecules to the semi-permeable membrane and it's the movement of water from a dilute to a concentrated solution is clear to you. Always remember, don't write it is the movement of substance from high concentration to low concentration. That's the wrong definition. It is high concentration of water to a low concentration of water is the correct one. Now, let's see what happens, how osmosis in plants takes place. Now, there are generally three kinds of solution. Hypertonic, hypertonic, isotonic. Hyper means more solute. So the hypertonic is that solution which has more solute, so less concentration of water. Hypotonic is the solution that has less solute, so more concentration of water. And isotonic is a solution which has same concentration of water in and out. So if we place a plant cell in a hypertonic solution, what happens? The outer solution has less water. So the water from the cell will move out of the plant cell by osmosis. So the plant cell will shrink and it will recede or remove away from the cell wall. And this is known as plasmolysis. So when a cell is placed in a hypertonic solution, it shrinks, and this condition in a plant cell is plasmolysis. On the other hand, when you place it in a hypotonic, that is more concentration of water, the water will move into the plant cell by osmosis. Then the plant cell will increase in size, become swollen. As it becomes swollen, it will give a pressure to the cell wall and the cell membrane. That pressure is known as a turgid pressure or the turgor pressure, and that makes the plant cell in its proper shape. On the other hand, if we place it in an isotonic solution, it will have the same concentration of water outside and inside. So there's no net movement of water. As a result, no pressure will build up and the cell will be soft. So in the plant cell, there are three terms: turgid, plasmolysed, and flaccid, and you need to remember these terms. Next is in plants, if you place it in a hypertonic solution, that is more concentration of solute, the cell will lose water by osmosis and shrink in size. If you place an isotonic solution, there's no net movement, so the cell remains intact. And in an hypertonic solution, the cell will gain water, so it will swell and it can eventually burst as well with time. Now, the next transport is the active transport. Active transport, as you remember, we did uphill and downhill. So active transport is uphill. That is movement of substances from low concentration to high concentration. That is against the concentration gradient with the use of energy. It is dependent on respiration as it requires energy. So the cell involved in active transport have lots of mitochondria. In plants, where active transport takes place, it takes place in the roots where water and minerals need to be absorbed against the concentration gradient so that maximum water and minerals can enter the plant root. And in animal, at times the digestive food, even after proper diffusion, need to be absorbed more so that the maximum absorption takes place. So their active transport plays a role. And in some of the marine organisms that has a lot of concentration of salts develop in their body, so they expel that excess salt by the Salt glands, which works by active transport. Okay, so I hope the movement of substances in and out of the cell is clear to you. Now we need to do the next thing, that we know the cell perform its function, but the cell has to divide. Why the cell needs to divide? For reproduction, growth, and repair. Now, how does the cell cycle takes place? This cell cycle is divided into 3 stages. First is the preparation phase, which is the interphase stage. It is the longest phase of the cell cycle, in which the cell grows in size and prepares the cell by making all the proteins and the enzyme needed for cell division. And the very important step that takes place in the interphase stage is a replication of DNA, where the DNA duplicates its content. After this preparation or the interphase stage is done, then comes the mitosis stage. Mitosis is a division of the nucleus, in which the parent cell splits into 2 daughter nuclei, which contains exactly the same number of chromosomes as the parent cell, so the daughter cells are genetically identical. And next is cytokinesis. It is a division of the cytoplasm, which takes place after the division of the nucleus. Now, what is mitosis? It is a type of cell division in which a parent nuclei divide to form 2 daughter nuclei with exactly the same number of chromosomes as that of the parent nuclei. The daughter cell produced are genetically identical to the parent, and we can call them clones. This division is important for growth, regeneration, and repair, and mitosis is also important in asexual reproduction. Now, how the stages of mitosis takes place, here's a diagrammatic representation of it. We started with, although there are 46 chromosomes, 23 pairs, but for diagram, we are starting with 2 chromosomes. So these 2 chromosomes first duplicated. Once they duplicated, they lies at the center through these spindle fibers. The spindle fiber shrinks and breaks them from the middle, so that each end now has 2 chromosomes copy, and then the nucleus divides. So we get 2 daughter nuclei with exactly the same number of chromosomes as the parent nuclei. Okay, so I hope this mitosis is clear to you. You should know what are the stages of the cell cycle, how the cell divides, what is the importance of mitosis, and what is the significance of mitosis. Okay, now let us see how the development of a human cell takes place. Now let us look at how the process of cell specialization takes place. Now, let me first explain you what is the entire life cycle of a living organism. Now, we have a male parent and a female parent. Now, both male parent and female parents have 23 pairs of chromosomes. This is what you need to remember. We have 46 chromosomes, 23 pairs, from which 23 has come from our mother and 23 from our father. Now, male cell, they undergo another division. We just saw mitosis. There's another division called meiosis, which produces half the number of chromosomes and is required to make the gametes. So the male parent makes a sperm or a male gamete, and a female parent makes ovum or a female gamete, and both has 23 chromosomes. Now, during fertilization, they both fuse to form a zygote, and this zygote now has 46 chromosomes. So the 46 or 23 pairs numbers is restored. Now the zygote divides into 2, 4, 6, 8 and many cell types and forms an embryo. Now at this stage, the entire cell is unspecialized. It has no function. We do not know which part of the cell will form the digestive system, which part of the cell will form the circulatory system. After that, the process of cell differentiation begins. Now this embryo, which cells have not yet specialized, are stem cells. So stem cells are the undifferentiated mass of cell which can differentiate into any cell type. Now after this, what happens is the process of cell specialization, where the cell becomes specialized to perform specific function. So some part of the cell will get specialized to form the circulatory system, some will specialize to form digestive system, and will give rise to a whole new organism. So in plant cell, the majority of the plant cell are the stores of stem cells. The root meristem, shoot meristem are the parts of actively growing part of the cell which contains the stem cells. We can take that and make a new plant. The plant can be grown easily because majority of the parts are not differentiated, and the ones which are differentiated, their differentiation is not permanent. So they can be reversed. Okay. Now in animals, majority of the cells are differentiated at a very early stage. In a plant, they get differentiated at a very late stage. So as in plants, they are differentiated at a very early stage. They are already specialized to form a nerve cell or a muscle cell. Now, adult cells replace the old and worn out cells in humans, but they also have a limited regeneration and the specialized power. And as majority of the differentiation is permanent, the cloning of the animals is difficult. So this is what happens in an animal differentiation, and this is what are the plant differentiation. Okay, so now, what are stem cells? So stem cells are the undifferentiated massive cells that can differentiate into any cell type. The sources of the stem cells are embryo, leftover remains of the embryo, umbilical cord. These are the sources of embryonic stem cells, and bone marrow is the source of adult stem cells. Now, what is the possible promises of the stem cells? There are a lot of problems in which the transplantation gets failed due to the rejection of the recipient from the donor, because the cell recognizes it as a foreign and the immune system tries to kill it. But if the stem cells of the person body are used to generate the organ, then the chances of rejection can be solved. Second, there are a lot of neurodegenerative disorder in which the part of the nerve cells get damaged. If the person's stem cells can recreate those nerve cells, and the neurodegenerative disease can be treated. The disease like blindness can be treated. It can be a cure of diabetes. If the beta cells of the pancreas get regenerated, it can help in a therapeutic cloning. In a therapeutic cloning, we take the adult stem cells, make it to make the embryonic stem cells, and then embryonic stem cells are specialized to make the desired organ. And it can also solve the organ damage problem. So stem cells is a very good promise, very good potential medicine revolution for future. However, there are a lot of issues against the stem cell. As the cells of the stem cells are rapidly dividing, we introduce the stem cell into an organism, it can lead to cancer. The stem cells can be contaminated and can cause unwanted diseases to the patient. The research in stem cell is still slow and expensive. The research happens on aborted embryos, which is considered as a potential source of life, and many religions have ethical concerns against it. And the knowledge of cell specialization, what genes get switched on and off, on differentiation is still incomplete. Okay, so I hope we have covered this topic in a much detail, and I have tried to cover the maximum of your specification. Now it's the time to test yourself by seeing if you know these key terms. So what are cells? Mitochondria, nucleus, cytoplasm, ribosome, prokaryotic, eukaryotic cell, cell wall, cell membrane, vacuole, microscopes, resolution, magnification, xylem, phloem, diffusion, osmosis, plasmolysis, turgid, flaccid, mitosis, differentiation, stem cell, therapeutic cloning. If you are still not clear with these terms, I would recommend you to go back, watch the videos, and then try over these key terms. So now you can pause the videos and have a go on trying these key terms. Okay, so I hope now you have written these answers on what is the meaning of these key terms. Now let us look at your answers. What are cells? Cell is a basic structural and functional unit of the living organism. Mitochondria is the organelle, which is the site of aerobic respiration. Nucleus is the organelle, which controls the activities of the cell. Cytoplasm is a jelly-like fluid which fills the cell and contain enzymes for chemical reaction. Ribosomes are the cell organelles for the site of protein synthesis. Prokaryotic cells are the primitive cell without nucleus or membrane-bound organelles. Eukaryotic cells are the advanced cell type with nucleus and membrane-bound organelles. Cell walls is the outer layer of the plant cell, which provides shape and support to the plant. Cell membrane is the outer layer that controls what goes in and out of the cell. Vacuole is an organ present in the plant cell, which has cell sap and makes the cell turgid. Microscope is a device that is used to see the objects which are not visible by the naked eye. Resolution is the ability to distinguish between two closely placed objects. Magnification is the ability to enlarge an object. Xylem is a transporting tissue that transports water and mineral. Phloem transport food. Diffusion, movement of substances from higher concentration to lower concentration. Osmosis is the movement of water molecule from a region of high concentration of water to low concentration across a semi-permeable membrane. Plasmolysis is the shrinking of plant cell when placed in a hypertonic solution. Turgid is the fully swollen cell which has gained water by osmosis. Flaccid is a soft cell due to no net movement of water. Mitosis is cell division that produces identical daughter cell. Differentiation is cell specialization. Stem cell is an undifferentiated mass. Now, what exactly is homeostasis and why is it important? Now, homeostasis is the process of maintaining the constant internal environment. Now, the question is, what is constant internal environment? Constant internal environment is maintaining a constant temperature, pH, blood glucose, and salt and water level. Now, when we maintain a constant temperature, that is known as thermoregulation. Maintaining salt and water level is osmoregulation. So, maintaining all these factors is what is homeostasis. Now, how this homeostasis is achieved? This is achieved by two systems working in your body, the nervous system and the hormonal system. Now, why this constant internal environment is important? This constant internal environment or homeostasis is important for the enzymes, as it is the enzymes which are controlling all the reactions and metabolism of the body, and they need optimum conditions to work. If the temperature increases or the pH level increases, it can change the activity of the enzyme and it can affect your body reaction. So it is very important to maintain the homeostasis. Now, whether it is a nervous system or the hormonal system, how it works, it's the whole hierarchy, how it works. Like in an organization, we have different levels. Like we have the lower levels and then the people at the higher level and then the people at the management level. In the same way, the nervous system is very coordinated activity of the body. Now, how it works, this flowchart is very important. First of all, there's a stimulus. What is a stimulus? Stimulus is any change in the external or the internal condition that makes your body to take any action or produce a response. For example, suddenly the temperature increases. So what you'll do, you will wear lighter clothes, you will switch on the fan. So you take some action because you want your body temperature to be normal. So first of all, there's always a change. So change could be change in temperature, pH, salt and water level. Now this change is detected by the body. Now who detects this change? These are the receptors. Now the change could be external and the internal. So the external receptors are your sense organs. So for example, there's a change in light intensity, the eye will detect. Change in sound intensity, your ears will detect. Change in temperature, your skin will detect. Or there are internal receptors in the brain, because at times what happens, the outer temperature is normal, but suddenly your body temperature increases. So there are changes in the internal conditions which are detected by the receptors in the brain. Now, when they detect the change, they send the message to the controlling center. The controlling center is the central nervous system, which composed of brain and the spinal cord, which takes the information from the receptors, process the information, decides the action, and sends the message to the effectors. What are the effectors? Effectors are the muscles of the gland, because your body, to control, to make the stimulus, to respond to the stimulus, they need to either take any action, that is to move any muscle, or they need to release a hormone. So the effectors are generally the muscles of the gland, which are directed by the central nervous system to take actions, and then they produce a response. For example, there's a change in blood glucose level. So your brain receptors will detect it, goes to the central nervous system. Central nervous system directs the pancreas to release insulin. Okay, so effectors in this case would be the gland. Now let us look at what are the three types of neurons that we find in the nervous system. The first type is the motor neuron, which takes the impulses away from the CNS, which was here. So motor neurons were sending the message from the central nervous system to the effectors. And if you see this, it has a long dendrite. So if you remember in the cell biology, we did that. It's a special cell. It has a long dendrite that receives impulses, and they transmit it through the long axon. And then at the end, there's a nerve ending. Then there's a sensory neuron, which takes the impulses to the CNS. It takes the message from the receptors to the central nervous system. It has a small cell body and the long axon, and then the dendroid is also extended, and at the end you have the nerve ending. Then you have the relay neuron, which are found in the CNS. That is, these are the neurons which are found inside the brain and the spinal cord. So they connect the sensory and a motor neuron. So they just have a big cell body, does not have a long axon, as the impulses do not have to travel a long distance, and they have the nerve ending. So you should know the difference between the three types of neurons, where we find them and what is their function. And if possible, you should know this diagram of the neuron and what are the labelings, what are the each terms, each things which are there in this structure. Next is the reflex action. At times you must have seen that you touch something hot, you immediately remove your hand, and after you have removed your hand, you realize that you have taken any action. So you take that action, which is automated without even you thinking, because your body receptors has already detected it and found that it's an emergency situation. And what do? They send the message to the spinal cord and not to the brain. So there's a stimulus detected by the receptors. The receptor sends the message via sensory neuron to the spinal cord, remember, not to the brain, and then the effectors and produces a response. So you can see here it's a spinal cord. It's like a head-shaped structure, and if you touch something sharp or hot, the message goes via receptors, via sensory neuron to the spinal cord. Spinal cord sends the message to the relay neuron. The relay neuron sends it to the motor neuron and to the muscle to remove the hand. So it is the automatic response of the body to a stimulus. In reflex action, the message from the sensory neuron is passed to the spinal cord instead of brain. The spinal cord sends the message to the effectors and produces a response. Example, knee-jerk reflexes, touching hot objects, sudden closure of the light with bright light. The advantages of the reflex action is it is rapid, it is quick, it is automated, instantaneous, without any conscious thought. Okay, so I hope this concept of reflex action, how it works, and the advantages are clear to you. Next we have is a synapse. Now, you know that the message in the neurons is transmitted by electrical impulses. But there comes a point, which is a junction between the two neurons. And this junction between the two neurons, or the gaps between the two neurons, is the synapse. And here the message is not transmitted electrically. It is transmitted by the chemicals called the neurotransmitters. So what we have is, at the end of one of the neuron, we have the vesicles which contains this neurotransmitter. Whenever the electrical impulses arrives, these vesicles are forced to fuse with the membrane, release the neurotransmitters into the synapse. This neurotransmitters is taken up by the receptors of the other neuron, and it produces electrical impulses on the other neuron. Okay, so you should know what is a synapse and how synapse works, and this diagram is important. Okay, so next we have the biggest controlling center of the body, which is the brain. So in the brain, you should know this is a hypothalamus. Then the main part is the forebrain, and then here you have the midbrain, and then you have the hindbrain, which is your cerebellum. The forebrain is cerebrum, and then you have this stalk, which is your medulla oblongata. Now the cerebrum, which is your forebrain, is responsible for your consciousness, memory, intelligence, and the language. The cerebellum, which is the hindbrain here, which is a round circle, it is responsible for muscle coordination and balance. And this long stalk, which is the medulla oblongata, is responsible for unconscious activities like the heart rate, the breathing rate, and the gut movement. So you should know the labeling of each of these parts along with the function. Now, the brain scan is where we do the magnetic resonance imaging and take the images of the different parts of the brain, and then we find if there's any clot, if there's any thrombosis, if there's any problem there with the brain, and then we relate this problem with the loss of function of the individual. Now, there are a lot of problems with the brain scan because the brain is complex. There's a skull which protects the brain. There are thousands of neurons, thousands of neurotransmitters involved. The function of the different parts is still not known, and due to the presence of skull, it is difficult for the drugs to reach the brain. So the brain damage is the biggest problem. Okay, therefore people are advised to cover and protect the heads while they are driving to make sure the damage to the brain is protected. Okay, next is the eye. This is the structure of the eye. You should know different labelings and the function of each of them, and it is very important. So the front part you can see here is the cornea. It's like a little bulge outwards, and the function of the cornea is to refract the light so that it can enter the lens properly. Then in front of the eye, you can see this is an iris. This is like a muscle that controls this aperture, which is the pupil. Okay, a pupil is nothing but it is the aperture which opens and closes depending on the light it enters. If the light is not that bright, the iris will retreat back, increasing the hole so that more light enters. If there's a bright light, then the iris will decrease the size of the pupil. Then you have the lens. The lens function is to focus the light on the retina. Then the lens is attached with the help of the ciliary muscles. It can contract and relax to change the shape of the lens. And there's a suspensory ligaments that's a support the lens onto the ciliary muscles. Then you can see there is a retina. Retina is the place where the image is formed, and then you have the choroid layer, which is the blood vessels that gives all the nutrients required to the eye, and there's a fovea, which is the part of the retina only with the cone cell, and then there is a blind spot. This blind spot is where the optic nerves enters the retina, and this optic nerve sends the message from the retina to the brain so that it can process the information and helps you to see. And then you have the fluid that fills the anterior part, which is the aqueous humor, and the posterior part is filled with the vitreous humor. This is the fluid that fills the eye and helps the eye to withstand pressure. Okay, so I hope all these parts along with this function is clear to you. Now, eyes do different changes so that it can see the far objects and the near objects, and these changes are known as accommodation. So for a distant vision, you can see that the ciliary muscles relax, making the suspensory ligaments tense, which in turn makes the lens thin. So you can see to see the far points, the lens has become thin so that the image is properly focused onto the retina, and the retina then can sense the message to a brain via optic nerve. But when you see the near point, the ciliary muscles, they are contracting, making the suspensory ligament too slack so that the lens become thicker so that the image can properly be focused on the retina. So you should know what changes takes place in the lens for distant vision and the near vision. Now there are a lot of defects of vision. At times people can just see the objects which are just at a short distance and cannot see the objects which are at the long distance. So there's a short-sightedness. And the long-sightedness. So in short-sightedness, which is myopia, the image falls, you can see, in front of the retina so that you cannot see it properly because the eyeball gets elongated. You can see that. So for that, what we do, we use a concave lens which properly refracts the light and make sure it falls on the retina. In hyperopia, you need to remember the image falls behind the retina. So the eyeball gets shortened and it is corrected by a convex lens. So you should know these both defects of vision. Now, there are other new eye technologies that have emerged. Now we can take contact lenses because it's not always feasible for the sports player or for the models to wear the spectacles all the time, the concave lenses or convex lenses. So in that case, we put the lenses just on the surface of the eye, which can be soft, silk, disposable, and they can be used by any person at any age. Then there's a laser surgery in which we change the thickness or the curve of the cornea so that it can properly refract and make the image at the retina, but it is generally done at the adults because at the growing stage the people can still have problems, so laser surgery cannot be successful. Then there's a replacement lenses where you either replace the faulty lens or insert the correct one in place of the faulty one. Now there are some damage risks because it's a surgery, so there are some damage risks to the eye that is involved. Okay, so I hope these all defects of vision and what are the treatments is crystal clear to you. Now we have seen the hormonal control. Now let us see the nervous control. So to remind you, we started with this concept of homeostasis, and we saw that homeostasis involves the nervous system and the endocrine system. The nervous system uses the system of the neurons, and the endocrine system involves the endocrine glands, which releases the hormones to bring the body back into its normal levels. Now, what is the difference between the hormonal and the nervous system is that the hormonal system releases hormones, which are chemical messengers secreted by the endocrine glands. They are secreted into the blood, travel to the target organ. The target organ has receptors, and hormones bind to the receptors and triggers a response, and it produces a slower but a long-term response. On the other hand, the nervous system uses a system of neurons which sends electrical impulses to produce a response. The message is transmitted via electrical impulses. The response produces localized, and impulses do not travel large distances, but the response is quicker but short-term. So you should know this difference between hormones and nervous, how it works, and which one is more quicker and effective. Now there's a master gland. The master gland is a pituitary. Why it is a master gland? Because it controls the other gland of the body. It controls the follicle stimulating hormone, which are responsible for management of menstruation and reproduction. Antidiuretic hormone is also controlled by the pituitary gland, which maintains the water level of the body. And thyroid stimulating hormone is also controlled by the pituitary, which maintains the metabolic levels of the body. Okay, so it's very important to know that pituitary is a master gland, and why is it a master gland. Now, to explain why is it a master gland, you should know what hormones it control and how it works. So pituitary controls the follicle stimulating hormone, which works at the ovaries and makes the female sex hormones estrogen, and that is responsible for the secondary sexual characters and the development of egg and the menstrual cycle. Thyroid stimulating hormone released by the pituitary works on the thyroid gland, stimulate the glands to release thyroxine, and the thyroxine affects liver and kidneys and controls the metabolism. And the antidiuretic hormones targets the kidney, which controls the water level by causing reabsorption of water. Adrenal gland releases adrenaline, which targets liver and heart and prepares the body for fight and flight. Testes releases the hormone testosterone, which targets the male reproductive organ and develops secondary sexual characters. Pancreas releases insulin and glucagon. The target organ is the liver, and the insulin decreases the blood glucose level, and the glucagon increases the blood glucose level. Ovaries releases estrogen and progesterone, which targets the female reproductive organs, controls the development of the egg, menstrual cycle, and develops secondary sexual characters. So these are some of the examples of the hormones with their glands, their target organs, and their effect, and you should remember them all. Okay, next we have is how the blood glucose level is maintained in the body, and this is an example of glucose homeostasis. Now, this works by the pancreas, and the pancreas releases two hormones called insulin and glucagon. Glucagon, you need to remember. Insulin lowers the blood glucose level, and glucagon increases the blood glucose level. So after meals, what happens? The blood glucose level suddenly increases. So what does pancreas do? It releases insulin. Now, insulin has these effects. It increases the permeability of the cells to take glucose, so that more glucose enters the cell, so that blood glucose level can go down. If the body does not require any more glucose, it converts all the excess glucose into a storage carbohydrate, which is a glycogen. It also converts the glucose into the fats and stops the breakdown of fat, so that all these factors can result in decrease in glucose level. So this is how the glucose metabolism works. When the blood glucose level rises, the pancreas releases insulin. The insulin does all these factors and makes the blood level, glucose level to be normal. On the other hand, if the blood glucose level is too low, then the pancreas releases glucagon, and the glucagon does all the things opposite to insulin. So you need to remember glucagon is the hormone which increases the blood glucose level, and glycogen is the stored carbohydrate which is made in the liver, where excess glucose is converted into glycogen.
Now, what happens if your body glucose level is not controlled? When the body glucose level is not controlled, it can lead to diabetes, and there are two types of diabetes, type 1 and type 2. Type 1 is insulin dependent, as in this case, the body does not produce insulin. Type 2 is insulin independent, because in this case, body do produce insulin, but your body is not responding to it. Your body becomes resistant to insulin. So in type 1, it is caused by damage to pancreas or by some autoimmune disorder or by accident. There's a damage to the pancreas. Type 2 is caused by poor lifestyle and diet. In type 1 diabetes, the person can be given insulin injections. In type 2, it is treated with lifestyle changes and a balanced diet. Type 1 is most common in young ages, where type 2 is common in obese people. Type 1 can be genetic, and type 2 is mostly environmental. In type 1, drugs might not be required. We just require insulin injections, whereas in type 2, there are some drugs are taken to make the body to respond to insulin. Okay, so you should know the differences between the type 1 and type 2. So what are the treatments for type 1 diabetes? Insulin injections. Remember, we need to take insulin injection. We cannot take insulin tablets, because insulin is a protein hormone, and protein can be digested in the stomach. The insulin goes to the blood, and it converts the excess glucose into the glycogen and control the blood glucose level. In type 1 diabetes, people are asked to take less carbohydrates, and if in case there are a lot of problems in managing that, pancreatic transplant, pancreatic stem cell transplant, or using stem cell to regenerate pancreatic cell are some of the future cures for maintaining type 1 diabetes. For type 2, balanced diet, regular exercise, weight management, taking drug to increase sensitivity of pancreas to insulin, or taking more insulin injections with the thought that increased concentration might increase responsiveness are the possible management techniques to maintain the type 2 diabetes. Okay, so you should know the difference between both the types of diabetes and how we control both of them. Now, this insulin maintenance, or the maintenance of water level, which we'll be seeing later, is an example of negative feedback. What is negative feedback? Negative feedback is when the level of anything rises above optimum, like the glucose concentration, the body tries to decrease it. And if the level of anything decreases below optimum, the body tries to increase it. So body is always doing negative or opposite to what has happened, and this is known as a negative feedback. For example, if the blood glucose level goes down, then the body releases glucagon that increases the blood glucose level. If the blood glucose level goes up, the body releases insulin to decreases the blood glucose level. Okay, so I hope this example of negative feedback and what is negative feedback is clear to you. Now you should know there's another hormone called adrenaline, which is a fight or a flight hormone. It is a stress hormone, emergency hormone. What effect it does to your body? It increases your heart rate, it increases your breathing rate, it increases the blood flow, it dilates the pupil, it diverts the blood flow away from the gut, and it increases the blood flow to oxygen to the brain, so that you become more alert and take quicker action. Okay, so you should know the effects of adrenaline. Now, next we have to do the human reproductive system, the male reproductive and the female reproductive system. The male reproductive system, it consists of a penis, there's a urethra, there is a bladder, there's a seminal vesicle, there's a prostate gland, and then there's a testis which produces the sperm, which is there in a scrotum. The sperms are released to these sperm ducts, and the seminal vesicles, it releases semen which mixes with the sperm, and it goes out through this urethra. And this male reproductive organ, testes, it releases a hormone called testosterone, which is responsible for secondary sexual characters. And the female reproductive system is the system of the two ovaries, and then there's a fallopian tube. The ovaries releases the egg into the fallopian tube, which travels into the uterus, and then there's a cervix and the vagina. And the female hormone is the estrogen, which is released by the ovaries, and that is responsible for the secondary sexual characters. Now, it's a very important hormonal mechanism in the body, which is a menstrual cycle, which is purely hormonal. Now, menstrual cycle is a cycle for 28 days. And in these 28 days, these are the changes that takes place in the body. So you can see this in this diagram. From day 1 to 5 is where the bleeding stage happens, which is the menstrual stage. There's a shedding of the uterus lining, and you can see all the hormones, they are at the lower level. From day 5 to day 14 is the egg maturation phase. So that is a follicular phase where egg is matured in the ovary, and the hormones responsible for the maturation and development of the egg is the follicle stimulating hormone. So you can see that the follicle stimulating hormone is started to increase in concentration. Then at day 14, the egg is released into the fallopian tube, and this egg is released, and this release of egg is caused by a hormone called luteinizing hormone. So you can see there's a sudden surge of this luteinizing hormone at the day 14. Now, after day 14 to day 18, there's a luteal phase in which there's a sudden increase in the progesterone and the estrogen hormone, which maintain. So there's an increase in progesterone and the estrogen level, because progesterone and estrogen are responsible in maintaining the uterus lining. So they maintain the uterus lining, they make it thick so that they can support the fertilized egg and cause the pregnancy. But if in between 14 and the 28 days, the egg is not fertilized, then the level of the progesterone suddenly falls, and it leads us to the shedding of the uterus lining, causing menstruation, and it starts with the day 1 to day 4. Okay, so you should know what are the changes that takes place in all these days, what is the phase, and what are the hormones that are involved. So what are the hormones that are involved in menstruation? These are pure hormones: follicle stimulating hormone, luteinizing hormone, estrogen and progesterone. Follicle stimulating hormone and luteinizing hormone are released by the pituitary, and they are required in the first 14 days. Day 1 to day 14, it's the follicle stimulating hormones which causes the maturation of the eggs in the follicle and stimulates the production of estrogen, and the luteinizing hormone causes ovulation. Then the estrogen is released, stimulated by the FSH, and from day 14 to day 28, it develops a uterus lining and stimulates more LH but inhibits FSH. And the progesterone is released by the empty egg follicle in the ovaries, and it is responsible for maintaining the uterus lining from day 14 to day 28 and prepare for pregnancy. It inhibits both LH and FSH, so no menstruation happens during pregnancy. Okay, so I hope these various hormones of menstruation are clear to you. Now, to prevent fertilization, there's a contraception method, and the contraception method, they either prevents the sperm to meet with the egg, or if the sperm meets the egg, they prevent the implantation of the fertilized egg into the uterus, thus preventing the pregnancy. Now, this can be achieved by the barrier method. The barrier method includes using diaphragms or condoms to prevent the sperm to meet the egg, or the hormonal method, which prevents the egg to mature or prevents the implantation of the eggs in the uterus, or the chemical method, which uses spermicide to kill the sperms, or the intrauterine device, which prevents the embryo from planting, or surgical method, which provides permanent contraception. Okay, so these are the various contraception methods, and they can come in a long note maker or a small question. So you should know what are the various contraception methods that we have. Next we have is how contraception works. So in contraception, there are 3 major techniques involved: the contraceptive pill, the intrauterine devices, and the surgical method. Now, what are contraceptive pills? The contraceptive pills contains mix of female hormones, estrogen and progesterone. We call them mixed pill. They prevent the release of FSH, so thereby they prevent the maturation of egg. They also make thick mucus in the cervix, which prevents the entry of the sperm. They also prevent the uterine lining development, so they prevent the implantation. Some pills, they just contain progesterone, and at times the contraceptive implant is also inserted. So there you don't have to take the pills regularly, and this implant slowly releases progesterone and maintains the uterus lining. Or the contraceptive patch is also used, which absorbs and mixes the hormones into the blood. They have these side effects like blood pressure, they have to be taken daily, and they cause changes in the menstrual pattern. On the other hand, intrauterine devices are the mechanical devices like copper T, which is inserted into the uterus. It releases copper ions, which are toxic to the sperms, and this device also prevents the implanting of the embryo into the uterus. And some of these IUDs, intrauterine device, they also releases progesterone, which works the same way as the progesterone pills, which prevents the release of FSH, preventing the maturation of egg. They make thick mucus in the cervix to prevent the entry of sperms, and they also prevents the uterine lining development, preventing implantation. But IUDs have side effects like infections or internal bleeding. Then we have the permanent contraception, which are mostly taken by the people who already have kids. They do surgical methods, which is vasectomy, which is the male sterilization, and tubectomy, which is the female sterilization. In vasectomy, the sperm ducts are cut and sealed so that sperms cannot enter the urethra, which prevents fertilization. And in tubectomy, the oviducts are cut and tied to prevent the release of egg, which prevents the fertilization. The side effects of this is it is permanent, so you cannot resume your fertility back. But with contraceptive pills, you can stop the pills and the hormone levels can be normal. And in the intrauterine devices, you can remove the device and then the fertility is restored. Now, there are a lot of infertility problems that happens in people. The first problem is the ovulation problem, where the eggs do not mature or the eggs is not ovulating. In those cases, the women are given fertility drugs, which are mix of FSH and LH. So follicle stimulating hormones causes the development of the follicles, and luteinizing hormone stimulates the ovulation. At times, there's a tubes that is faulty, so that the embryo is not able to come to the uterus, or there's an implantation problem. In that, we perform an in vitro fertilization where embryos develop outside and it is implanted back into the uterus for the development of the pregnancy. And in the third case, even implantation happens, but pregnancy development doesn't take place. In that case, there are problems with the uterus. So the surrogate mother, where the fertilized egg is implanted into another mother who gives birth, can be a solution. Now, what is in vitro fertilization, which is very promising, and that has given babies to many of the people who do not have babies. So what do we do is, we first give fertility drugs and stimulate ovulation. Then we collect the egg and the sperms, and we put it into a petri dish, and we cause the fertilization to occur. When the fertilization is occurred, we cause the zygote to divide and develop into the embryo. The embryo is then taken and inserted surgically into the uterus lining. Okay, and then this develops into a baby. So this is what is the concept of in vitro fertilization, but it has problem as it is expensive. It can result into multiple embryos, it can result into premature birth, it can result into birth with disability, and it cannot be always successful. But that has caused, it has shown a good success rate, and there are a lot of people who are opting for in vitro fertilization. Next is plant hormones. As in humans, there's a hormones controlling the body mechanism. In the same way, plant also needs some survival changes. For example, they should grow towards light, the root should go towards the gravity. So these are controlled by the plant hormones. So there are two important movements that we see in the plant is phototropism, where the shoot moves towards light. You can see in this example, the shoot is moving towards light, and there's a gravitotropism where the root moves towards the gravity. Now, why the shoot moves towards light? It is caused by auxin. When auxin is produced in the root tip and the shoot tip. So when the light falls on the auxin, you can see auxin has shifted towards the shader side. And when the auxin shifts towards shader side, auxin causes the growth of the cell elongation of the shader side. As a result, cell divides onto the shader side, causing the shoot towards bend towards light. On the other hand, in response to gravity, the auxin shift towards the grounder side. So towards the lower side, the auxin develops in response to gravity, and in the roots, auxin has an opposite effect. Where it is concentrating, it is inhibiting the growth. So the other side of the root grows, which results in the bending of the root downwards. So auxin causing bending of the root downwards, and it causing the shoot to grow upwards. Why? Because Then phototropism, auxin is causing the growth in the shader region, and in the gravitotropism, the auxin is inhibiting the growth onto the side where it is present. So you should know how both of these mechanism works. Now there are 5 important hormones in the plant which are very important: auxin, gibberellin, ethine, cytokinin, and abscisic acid. Auxin stimulates cell division and is responsible for growth of the plant. It is used in the tissue culture as a rooting powder, as it causes the root to develop quickly. And it is also used as a weedicide, because when the weed, due to its broader leaf, take the auxin quickly as compared to our main crops, and these concept of thermoregulation as what changes takes place in the body when suddenly the temperature becomes too low. So when the temperature becomes too low, you must have seen the body starts to shiver. Why? Because the body detects that there is a sudden decrease in the body temperature, so the heat needs to be generated. So shivering increases the heat so that the body temperature can raise to normal. Second, the thyroid stimulates cells to increase the metabolism so that the heat production can be increased. You must have seen you get goosebumps in cold. Why? Because the body hair stands up so that they can trap the layer of insulated air, which can prevent heat loss by conduction. And you must have seen your body tends to get blue. Why? Because of the vasoconstriction. So when the temperature becomes too low, the capillaries that is supplying the blood to the surface of the skin, they constrict. They reduces the blood flow to the surface. As the blood flow to the surface is reduced, the blood can lose less heat due to radiation. On the other hand, when it gets too hot, the body hairs lies flat. There's a sweating. The sweat glands produces sweat. The sweat causes cooling of the body by evaporation. The metabolism of the body decreases. So the thyroid stimulates cells to decrease metabolism so that the less heat can be produced. And then there's a vasodilation. What is vasodilation? The capillaries that are providing the blood vessels of the capillaries that supplies the blood to the surface dilate so that more blood flows to the surface. And when more blood flows to the surface, it will lose heat by radiation. So in vasodilation, or in the hotter times, we are making more blood flows to the surface. So you should know what is the difference between vasoconstriction and vasodilation, and what changes takes place in the body in response to temperature. Okay, now let us see what are the different waste products produced in the body. Waste products are the products which are produced by the metabolic reactions like respiration and digestion. The first waste product is carbon dioxide produced during respiration. It is toxic because carbon dioxide can disturb the pH and affects the enzyme activity. So it needs to be removed immediately. So it is excreted out through the lungs. Then there are water. Then by metabolism, we also produce water, like in respiration or during the digestion process. It is excreted through the skin in the form of sweating or breathing, or kidneys in the form of urine. Water can also disturb the osmotic balance and salt levels. So it is very important the level of water is also maintained in the body. Now, when there's excess carbohydrates, excess fats due to digestion, that can be easily stored in the body. But protein is very harmful. Excess protein cannot be stored in the body. So the protein is digested into amino acid, and the liver deaminates. That is, by the process of deamination, converts amino acid into ammonia, and ammonia is converted into urea. So urea is another waste product produced in the body by the metabolism of protein, and this urea is excreted by the kidneys in the form of urine. Okay, so how these kidneys function? So we have 2 kidneys, and these kidney receives the blood. They filters the blood, and they pass the urine into via these 2 tubes called ureters into the bladder. The bladder stores the urine and expels the urine out through urethra. Now there are 2 systems that works in the excretory system: the ultrafiltration and the selective reabsorption. In ultrafiltration, the kidneys filters the blood at a very high pressure. All the waters, glucose, and useful components get also into the kidney filtrate. The blood cells and the blood proteins are too big size to be filtered. Now, once these essential things are there into the kidney filtrate, they need to be reabsorbed back into the blood. So this process is known as selective reabsorption, where the useful components are reabsorbed back into the blood. The waters get reabsorbed at different levels depending on need of the body. Therefore, we call it a selective reabsorption. Now let us see how the body regulates the water level. And the regulation of water level is known as osmoregulation, and it is also a negative feedback. And it involves the release of a very important hormone called an antidiuretic hormone by the pituitary. So when the water level decreases in the body, it is detected by the osmoregulatory centers in the brain, which directs the pituitary to release a very important hormone called the antidiuretic hormone, which travels into the blood and enters the kidney tubules, and it increases the reabsorption of the water into the blood by increasing the permeability of the kidney tubules. As a result, more concentrated urine is passed, and it also promotes thirst, so that you take in more water to resume the blood water level. On the other hand, when the water level increases, it is also detected by the osmoregulatory centers in the brain. The release of ADH in this case is inhibited by the negative feedback. So there's less reabsorption of water, so more dilute urine is passed. So you should know how this mechanism of osmoregulation works. Now, there are people whose kidney get damaged either by an autoimmune or a genetic disorder or by some accident. So in this case, they have two solutions. First, either they take the help of the artificial kidney called the dialysis, or they do a kidney transplant. Now, what happens in a dialysis is the patient blood is taken out and it is given a heparin, which prevents the blood clotting, and then the blood is fed into a dialysis fluid, a dialysis machine which has a dialysis fluid. The dialysis fluid contains no concentration of urea and the ideal concentration of minerals and the salt level as that of the blood, and the dialysis tubes are made up of the semi-permeable membrane. So as the blood flows into the dialysis fluid, the urea is diffused out along the concentration gradient, and excess salt is also removed, maintaining the normal salt and mineral level and absorbing all the urea into the dialysis fluid. The blood now becomes pure and clean and is pumped back into the patient. Okay, so in the artificial kidney, the blood flows into the dialysis machine, which contains the dialysis fluid. It contains the same concentration of essential mineral ions as that of the blood, but no urea. So as the blood flows into the dialysis fluid, the urea is diffused out along the concentration gradient, and excess salt is also removed, maintaining the normal salt and the mineral ion level, and the clean blood is then pumped back. The problem with the dialysis is that you need to regularly visit the hospital, you need to have the lifestyle changes, the changes diet and regular expenditure are some of the disadvantages. On the other hand, there's a kidney transplant. You can see there's a damaged kidney. So from a donor, a normal kidney is transplanted, and that works normally as like of the kidney. So the donor has to be a close match, because the body can reject the donor kidney, because it can take that as a foreign antigen and produce antibodies and destroy it. So what do we do in this case is the person has to be given an immunosuppressant drug so that the body immune system does not reject it. But the problem is it does not last longer, and since the person is on immunosuppressant, the person can get prone to infectious diseases. Okay, so there is a trade-off. So which is the best way, dialysis versus kidney transplant? It depends on the patient condition, what he is willing and what he's not willing to do. Because dialysis has an advantages of getting no surgery, no infection, no immunosuppressant drug, and easily available. But the disadvantages of lifestyle changes, regular visit, restricted diet, and a long procedure. On the other hand, kidney transplant does not require any lifestyle changes, no diet restriction, no regular visit, but it does not last longer. There are chances for rejection. There are immunosuppressant drugs need to be taken. The person is more prone to infection, and finding a suitable donor is a big problem. So the promising future for the kidney disorders is using stem cells, the person's own stem cells, to create the kidney cells or the nephrons, so that the problem of rejection can be avoided and it can be a long-term solution. But again, the development and the research is still going on. Okay, so I hope this topic A long topic with a lot of key terms is crystal clear to you. Now let us see what are the key terms you should know. So you need to write about all these key terms, pause the video, write about these key terms and come back. So you should know what are homeostasis, receptors, effectors, stimulus, neurons, central nervous system, sensory neuron, motor neuron, relay neuron, reflex arc, brain, cerebral cortex, cerebellum, medulla, MRI, eye, cornea, retina, blind spot, ciliary muscle, suspensory ligaments, sclera, iris, pupil accommodation, myopia, hyperopia, endocrine system, hormone, adrenaline, insulin, pituitary gland, FSH, LH, estrogen, progesterone, glucagon, glycogen, diabetes, menstruation, ovulation, IVF, phototropism, gravitotropism, oxygen, jubilin, ethene, vasoconstriction, vasodilation, thermoregulation, dialysis, and selective reabsorption. Okay?
Now, in the previous unit, we studied about the cell, which is the basic structural and the functional unit of the body. And the examples are the nerve cells and the muscle cells. Now, these cells, when the group of cells combine to perform a specific function, that becomes the tissues. When groups of tissues combine together to perform a specific function, it becomes organs. And when groups of organs combine together to perform specific functions, that become organ system. An organism is nothing but it's the collection of different organ systems. Now, what are the different organ systems? Like circulatory system, digestive system, nervous system, excretory system, they make up the different organ system of animals. And if we talk about just the digestive system, the different organs in that will be stomach, kidney, liver, pancreas, heart. These are the different organs. And then the organs are further made up of smaller tissues like muscular tissue, epithelial tissue, glandular tissues, and the tissues are made up of collections of cells. So you should know these hierarchy and the examples of them. Now, let us talk about digestion. Now, what is digestion? Digestion is the process of breaking down of large insoluble food particles in the food into soluble smaller particles which can then be absorbed in the blood and used up. Now, what are the bigger components which are present in the food? These are carbohydrates, proteins, and fats. The carbohydrates get digested by the special enzymes called carbohydrases into simple sugars. Proteins get digested by proteases into amino acids, and fats are digested by lipases into fatty acids and glycerol. Now, this is what is the structure of the digestive system, and you should know how to label it. So it starts with the mouth and the pharynx, and then there are the salivary glands that releases saliva which softens and moistens the food, and saliva also contains the enzyme amylase that begins the process of digestion of starch to sugars. So you must have seen people say that if you chew the food, it tends to taste sweeter. The reason is that the amylase enzyme, it starts to digest the starch into the food into simple sugars. Then the food is pushed down into this food pipe called esophagus, and then it reaches the stomach. In the stomach, the hydrochloric acid is released. This hydrochloric acid first kills all the infections which comes up with the food, and it also makes the pH acidic so that stomach enzymes can work, because stomach enzymes work in the acidic pH. The digestion of protein starts in the stomach. The food stays approximately 3 to 4 hours in the stomach, and then it goes into the small intestine. When it goes into the small intestine, it gets two secretions. One, from the gallbladder, it receives the bile. Bile is made in the liver and stored in the gallbladder. The bile, it emulsifies the fat and neutralizes the stomach acid pH, which comes up with the food. And then the pancreas releases the pancreatic juice, which contains all the enzymes to finish up the digestion. So all carbohydrates, proteases, and lipase are present in this pancreatic juice. The digestion is finished. In the small intestine, the food gets absorbed into the blood, and then undigested food passes into the large intestine. The large intestine absorbs all the water and converts the undigested food into the semi-solid mass called the feces, and the feces is thrown out of the body through rectum or the anus. So this is the whole process of digestion. If you see this, we can break this digestion process into 5 stages. One is ingestion. Ingestion is the taking in food. It helps with the help of the mouth and the tongue. The food is churned, so mechanical digestion takes place, and the food is mixed with the saliva. Then is the digestion. It starts in the mouth, and stomach, intestine, pancreas, and liver are involved in it, which is the breakdown of large insoluble particles into smaller ones. Then in the small intestine, absorption, the process of digested food are absorbed into the blood. Then assimilation happens in the body cell, where this absorbed food is used for the body processes. And there's an ingestion, egestion, which is the getting rid of undigested food material, which happen through rectum and anus. Okay, now let us just quickly summarize this entire digestion process. So it starts with the mouth, where food is ingested, mechanical digestion starts, and amylase is mixed, which starts to break down the starch into the sugars. Then it is the pharynx, which is at the beginning of the throat, and both food and air travels to it. Then there's a small flap called epiglottis. Epiglottis is a flap of muscles that closes the windpipe so that the food does not get into the windpipe, and it directs the food into the esophagus. Esophagus is the tube that leads the food into the stomach, and it has some peristalsis movement that pushes the food down. Then the food comes into the stomach, where the mechanical digestion and the chemical digestion starts. The hydrochloric acid is released, and an enzyme called pepsin is released that digests proteins into amino acid. Then the food goes into the small intestine. There, the liver that makes the bile stores into the gallbladder. The secretion of the bile comes into the food that neutralizes the food, and then gallbladder also releases bile, which is stored in the gallbladder. And then the small intestine, where the digestion finish and the absorption starts. And then there's a small organ here called the pancreas, which releases the pancreatic juice. And yeah, pancreas, it is where it produces the pancreatic juice that contains the lipases, proteases, carbohydrates enzyme. Then the food goes into the large intestine, which absorb all the water. Then it is thrown out of the body through the rectum and anus. So you should know all the digestive organ. You should be able to label them in the exam, and you should know the function of each of the parts. Okay. Now then, the very important function is the bile juice. It is a greenish yellow alkaline liquid which is produced in the liver and stored in the gallbladder, and it performs two major functions. You should remember that. One is the neutralization. So the food that comes from the stomach is acidic, and the enzyme of the intestine can only work in alkaline condition. So the bile neutralizes the food that comes from the stomach and makes it alkaline, so that enzyme release in the intestine can work effectively. Next is the emulsification of the fats. For lipase to work, the fat must be broken down into smaller droplets to increase the surface area for the lipase to function. The bile performs this function of emulsification of fat for the efficient working of the lipase. Now, let's see what are the components that we take in with the food. We take carbohydrates, proteins, and fats. Carbohydrates are present in bread, pasta, cereals, and rice. Proteins are present in pulses, chicken, meat, poultry, egg, beans, and nuts. And fat is present in butter, oil, margarine. Carbohydrates are composed of sugars. Proteins are made up of amino acids, and fats are made up of fatty acids and cholesterol. And this is what these components get digested into. The enzyme for carbohydrates are carbohydrates. Example, amylase. For protein, it is proteases, and for fat, it is lipases. Carbohydrates are digested in mouth and small intestine. Proteins are digested in stomach and small intestine, and fats are digested just in small intestine. Carbohydrates are the principal source of energy. They are fuel for respiration. They can be storage molecules like starch and glycogen, or structural molecules like cellulose in plants. Proteins are the components of muscles required for growth and repair. They're the components of enzymes, and they are also components of some of the hormones like insulin. Fats are used to insulate the body. They are the reserve source of energy, and they also makes the cell membrane. Okay, so you should know what are the different components of food, their sources, where they're digested, and what is their function in the body. Okay, so now since we are doing digestion, and digestion is chemical, that is, it involves the use of enzyme, you should know what are enzymes. So make sure you remember this definition and this lock and key model, very, very important. So I'll just take you through it. So enzymes are biological catalysts. So I think in chemistry, you remember what are catalysts? Catalysts are those substances that increases the rate of reaction without being used up. So if there's a catalyst which are used in biological reaction, we call them enzymes. So enzymes are biological catalysts that increases the rate of a biological reaction without being used up. Now, what do enzymes do that increases the rate of reaction? They actually provide an alternative route to a reaction, which works by lowering the activation energy. And enzymes are chemically proteins, so they are very sensitive to heat, pH, and temperature. Okay, now this is the structure of the enzyme, and the enzyme has a specific shape, and it has a site called the active site. Active site is a site where substrate molecules bind to it. Now substrate and the active sites, they have complementary shape. Remember to use the word complementary. Most of the time in the exam, students say substrate and active site have same shape. No, that's wrong. It's like a lock and key. The substrate and the active site, they have a complementary shape. The substrate binds to the active site, forms an enzyme substrate complex. The enzyme substrate complex react to form an enzyme and the product, and it disintegrate to give the product back, and your enzyme gets regenerated. So remember, enzymes are highly specific due to the active shape. As the active shape has a specific shape which is complementary to the substrate, so the specific substrate molecule can fit into the active site of the enzyme. Now, what are the factors that affects the enzyme activity? Temperature and pH are one. We'll be discussing that. But the substrate concentration and enzyme concentration also affects the rate of reaction. So as we increase the substrate concentration, there'll be more molecules to bind with the active site. Therefore, the rate will increase at the start. But after that, it levels off because all the enzyme active sites will be used up, and then enzymes will become a limiting factor. The same thing work with the enzyme concentration. As we increase the enzyme concentration, there'll be more active sites for the substrate molecules to bind to. But after that, further increasing the enzyme concentration will have no effect because then all the substrate molecules will be used up. So substrate will become limiting, so the graph will level off. Now for the temperature, as we increase the temperature, the rate of enzyme catalyzed reaction increases because the particles gain kinetic energy as per chemistry. They'll collide more the collision, more the chances of reaction. It increases up to a point, and this point is called the optimum. Optimum is the point where the enzyme activity is the highest. But after that, the rate will drop suddenly. Why is that? This is because the enzyme gets denatured. Now, what is denaturation? Now, denaturation is when you heat any specific enzyme due to being protein in nature, their bonds break. So they change the shape. Now, when they change the shape of the active site. So the substrate molecules can no longer bind to it, decreasing the rate of reaction. So denaturation is change in shape of the enzyme with a change in pH or the temperature. The same effect happens with the pH. As you increase the pH, it increases the rate of reaction up to the optimum, and after that it drops. It doesn't drop that suddenly like temperature, so we make like a bell-shaped curve. And then it again drops. Why? There's a denaturation because the shape of the enzyme changes. Now for the temperature, the optimum is around 37 to 40 degrees because that is the normal body temperature. But for the pH, different enzyme has different optimums. Like stomach enzymes, they work best in acidic condition, and the intestinal enzymes, they work in the alkaline condition. Okay, so I hope this graph is clear to you. You should be able to draw this graph in the reaction in the exam, or if this graph is given, you should be able to explain all this trend, why there's an increase, what is an optimum, and why there's a drop. And I hope you will remember it. Now let's move on to a circulatory system. The question is, why we need a circulatory system? And what is a circulatory system? So circulatory system is a system that provides the body with oxygen, nutrients, hormones, or all the essential things that a body needs to survive. And it also gets rid of the waste products. But why do we need a circulatory system? We need a circulatory system because we need constant supply of all these nutrients. And circulatory system consists of three components: a pumping organ, an organ that pumps the transporting system, transporting medium. Transporting medium is the blood, which has all the things that a cells required. It is pumped by a pumping organ called the heart, and the blood is taken from one cell to another, or from one organ to another, to the system of vessels, which is arteries, veins, and capillaries. So now we'll be looking over what is blood, heart, and these arteries, veins, and capillaries. Now, if you talk about the blood is made up of blood plasma and blood cells. So if you see blood, we centrifuge the blood. Then the blood cells are heavy, they collect at the bottom, and then there's the blood plasma. Blood plasma makes up 55% of the blood, and it is the liquid component of the blood, and it has all the things that a cell requires, like glucose, amino acids, hormones, waste product, carbon dioxide. And then the blood cells, they perform specific function, and there are three types of blood cells: red blood cells, white blood cells, and platelets. The function of the red blood cell is to transport oxygen all around the body, and it's a special cell. Why it's a special cell? Because to carry out these special functions, it has special adaptations, like they are in the form of biconcave disc, which increases the surface area for oxygen transport. They have no nucleus, so that they can have more room for the oxygen, and they contains a pigment called the hemoglobin, which binds with oxygen and carry it all around the body. Then you have the white blood cells. White blood cells are the soldiers of the cell. They protect the body from infections. They are made up of two types of cell: lymphocytes, they produce antibodies, and phagocyte, which engulfs the pathogen. And then we have the platelets. They are involved in blood clotting. They have fibrin proteins which form mesh around the blood and prevents the blood from excess blood from flowing out by forming a clot. So I hope you will remember what are the different cells and their function, and what is the function of the blood plasma, and what are the different components of the blood. Okay, so now we will talk about the heart, but before that we should know why we need a circulatory system. We need a circulatory system because we have low surface area to volume ratio, we are metabolically active, and we are multicellular. Now, low surface area to volume ratio because we have a bigger size. As the size increases, the surface area to volume ratio decreases. And at such a low surface area to volume ratio, diffusion alone is not effective to meet the growing need for the body. Therefore, we need a circulatory system. Now, being metabolically active, we need a constant supply of oxygen, glucose, and nutrients. And at the same time, the waste product is also made at a greater level. So we need a constant removal of waste products. So we need a circulatory system to do that task. And being multicellular, we have lots of cells which are deep inside. So diffusion distance is too large. So we need a circulatory system to transport substances deep inside the cells as well. So therefore, we have this circulatory system, and the circulatory system we have is double circulation. Now, let me explain you how this double circulation works. Now, in our circulation, we have a lungs and the heart is 4 chambers. The top chambers are the atriums, and the bottom is the ventricle, and our right is the heart left. So this is the left atrium, right ventricle, right ventricle, and the body. Now, how does the circulation start? The lungs gives oxygen to the blood, and the oxygenated blood from the lungs, it starts flowing to the left atrium. The left atrium then pushes the blood to the left ventricle, and then left ventricle pushes the blood out, and it goes to all parts of the body. The body takes oxygen and all the required nutrients from the blood, and then it releases carbon dioxide into the blood, and the blood becomes deoxygenated. This deoxygenated blood from the body is pumped back into the right atrium. The right atrium takes the blood to the right ventricle, and the right ventricle then takes it back to the lungs so that it can pick some more oxygen, and the cycle continues. Okay, now if you see the circulation, it is consist of 2 circuits. One, lung to the heart and heart to the lung, and that is known as pulmonary circulation, and heart to the body and body to the heart, that is known as systemic circulation. And both of these circulation together make a double circulation. So that is one of the definition of double circulation, that it consists of 2 circuits, a pulmonary and the systemic. The second way to describe this double circulation is, this is circulation in which the blood is pumped to the heart twice in one complete cycle. So in one complete cycle, the blood is pumped to the heart twice. It involves 2 circulation, pulmonary, which is between lungs and the heart, and systemic, which is the circulation between heart and the body. The double circulation makes the circulation more efficient by preventing the mixing of oxygenated blood, and helps to alter the pressure from the different chambers of the heart. So here you can see in double circulation, the oxygenated blood is all to the left side and deoxygenated blood to the left side, and the heart can modulate the pressure. When it is sending the blood to the body, it can increase the pressure, and when it is sending the blood to the lungs, it can decrease the pressure, and that can only be done as it has a double circulation. Now let us label the vessels. Now, if to label the vessels, always remember anything that gets the blood to the heart will be the vein, and anything that takes the blood away from the heart is an artery. So in the first case, the blood is coming to the heart from the lung. So this has to be a vein. And since it is the vein associated with the lung, so it has the word pulmonary vein associated with it. So the blood comes from a pulmonary vein to the left atrium. Then it goes to the left ventricle, and then from the left ventricle, it goes to the body. So blood is going away from the heart. So this has to be an artery, and it has to be a largest artery, as it is giving the most difficult task of sending the blood to all parts of the body. So this is aorta, which is our largest artery. Then the blood comes from body to the heart. Again, it has to be a vein. So it's a vena cava. And then from the right ventricle, it goes back to the lung. So it has to be the artery. And since it is associated with the lung, so it is the pulmonary artery. So you should be able to draw this circuit and label all the vessels. And I hope this is clear to you. Next is we have this heart. Heart is a pumping organ, works all day and night. It has 4 chambers. Atriums are the top. So this is left atrium. This is right atrium. The ventricles are at the bottom. So this is left ventricle, the right ventricle. Ventricles are doing the biggest job as they are pumping the blood to a greater distance. So you can notice that atrium has a thin wall and ventricle has the thickest wall. And as compared to left ventricle and right ventricle, left ventricle has to send the blood to all parts of the body. So it has to send the blood at a very high pressure. So it has the thickest wall. Okay, and then the left atrium receives the blood from the pulmonary vein. It goes to the left ventricle. The left ventricle sends the blood to all parts of the body. So that's the aorta. The blood travels all parts of the body and come back to the right atrium through the vena cava, and from the right ventricle, the blood pump back to the lungs via pulmonary artery. So you should know the structure of the heart and the labeling and the functions of all the parts, and I hope this is clear to you. Now, if you remember that in a circulatory system, we need the blood vessels that carries the blood all around, and there are 3 types: arteries, the capillaries, and the veins. The capillaries connects arteries and veins, and what you can see in arteries and vein, the lumen of the vein is bigger and the lumen of the artery is smaller. On the other hand, the artery is surrounded by a thick muscular tissue which is not present in the capillaries, and veins have thin. So let's tabulate the difference between them and make sure you remember all these differences and able to write in the exam. Arteries carries the blood away from the heart. Veins carries the blood to the heart. Capillaries connect arteries and veins. Arteries, blood flow at a high pressure. In veins, it flow at a lower pressure, and capillaries flows at a lower pressure. So to withstand high pressure, the arteries have thick muscular elastic wall. Veins have thin walls, and capillaries are 1 cell thick. Arteries have a narrow lumen, veins have a wider lumen, and capillaries have a very small lumen. As the blood flows at a very low pressure in veins, there are chances that the blood can backflow. So veins have valves to prevent the backflow of the blood. Arteries and capillaries do not have valves. Arteries generally carry the oxygenated blood except the pulmonary arteries. Vein carries deoxygenated blood except the pulmonary vein. And the capillaries carries both oxygenated and deoxygenated. So I hope you were able to spot by the diagram whether it's in arteries, veins, and capillaries with their function. Okay, now since we have discussed the heart, there are a lot of problems that can come up with the heart, and they are known as coronary heart diseases. Now, what are coronary heart diseases? If there's a problem in a coronary artery. What's a coronary artery? It is an artery it's the branch of the aorta that supplies the blood to the heart. And if these arteries become narrow, why they become narrow? You can see this is a normal coronary artery, and in the coronary artery there is a plaque that is deposited. What is a plaque? It is a fatty materials like cholesterol which get deposited. It narrows the artery. The narrowed artery reduces the blood flow to the heart. The blood flow to the heart is reduced. The heart do not get enough oxygen. It can cause heart pain, chest pain, and heart attack. So this is what is the plaque deposition that happens in a coronary artery. Now, how we can improve that? We can insert a stent. A stent is placed which has a balloon with it into the blocked artery. The balloon is inflated and the balloon opens the blocked artery, and a mesh stent made up of metal is placed which holds the narrow artery and prevent it again from collapsing, and people are also given the medicine called the statins, which lowers the blood cholesterol and prevents the further deposition. So this is one of the way, and this is also known as angioplasty, in which a balloon or the stent is used to open the blocked artery. But there are cases where the stent cannot be performed due to maybe the blockage is too much, or some of the problems to be done a patient can have with the stent. Then in that case we do a bypass. What's a bypass? See, you can see that this is a blocked artery. We give a graft, and this graft bypass the blocked artery. Okay, so there's a blockage in the coronary artery, and artery graft is stitched to the coronary artery, so the blood, rather than flowing through the blocked artery, it flows to the graft and it resumes the blood flow. Okay, now there are another problems as well apart from this that can be associated with the heart. These problems are like a leaky valves. A person can have leaky valves, maybe by time that the blood flows at a very high pressure, so the person can develop leaky valves. So due to increased pressure of blood flow, the valve starts to leak. For that, we have valve replacement, where the valves made up of metals or polymers are placed, or the biological valves from the animals can be used. At times, the person get irregular heartbeat. Why? Because the heart is controlled by the groups of cells present in the left atrium called the pacemaker, which regulates the heartbeat. If there's any problem with that, there's an irregular heartbeat. So we make electrical devices called artificial pacemaker, which takes over the function of the natural pacemaker, and they regulate the heartbeat. Then, apart from all this problem, the person can have a greater damage to the heart or heart failure. In that case, we need a heart transplantation or artificial heart, which is not that successful, but the research is going on, but heart transplant can be done. So these are all the non-communicable diseases, or you can say the problems with the heart, and the proposed treatment for the same. Okay, now after the circulatory system, we come to the human gas exchange system called the respiratory system, which starts with the nose, and then the air goes into the windpipe. The windpipe is known as the trachea. The trachea is covered by the ring of cartilages that prevents the trachea from collapsing, and then the trachea bifurcates into two. These two bifurcations are known as the bronchus. These two bronchus enters the lungs. In the lungs, the bronchus gets further branched into the bronchioles, and at the tip of the bronchioles, we have greater air sacs called the alveoli, where the gas exchange takes place. And then there's a dome-shaped muscular organ called the diaphragm, which goes up and down to regulate the chest volume for inspiration and expiration. Now, what is the function of the respiratory system? Breathing system is to breathe. Breathe is taking in oxygen and giving out carbon dioxide. Now, for the air to come in, air always move from high pressure to a low pressure. So since the air coming in, the pressure inside the lung is low. For expiration, the pressure inside the lung is high. If the pressure is low, the volume of the lung should be high, and the volume of the lung should be low in expiration. How these volume increases in inspiration? It is the muscles, the ribs muscle, they contract and the rib moves outwards. In expiration, the ribs relaxes and the rib move downwards. The diaphragm, it flattens, which increases the volume and the air moves in, and expiration, the diaphragm domes up, which decreases the volume and the air pushed out. So you should know what is the mechanism of inspiration and expiration, and what changes takes place inside the lungs or the ribs for inspiration and expiration to take place. For gas exchange, if you remember, we talked in cell biology, there's an alveoli which has a greater surface area by having a special shape and being millions of them. They have a shorter diffusion distance by being one cell thick, and they maintain a steep concentration gradient by having a rich blood supply. By all this factor, alveoli has adapted themselves for an efficient gas exchange. Now we have discussed organization in animals. Now let's discuss organization in plants. The plant cell is there. We discussed the plant cell in the cell biology video. Then the plant cell get organized into tissue, xylem, phloem, these palisades and spongy mesophils. These are the tissues. You can see sponges are the ones which have greater air spaces between them but are photosynthetic. The palisade cells are arranged next to each other, which are also photosynthetic. In the organ system, we have a water transport system and the food transport system. Water transport system consists of xylem and the roots. The food transport system consists of the phloem and the leaves, and all of this make a plant organism. Okay, now what is a transpiration? Transpiration is the loss of water from the surface of the leaf in the form of water vapor, and this happens to a stomata. So stomata are these air spaces or air pores present on the surface of the leaf, which consist of stomatal pore and these cells, which are known as the gut cell. The gut cells open and closes, which controls the water movement, and the gas exchange also takes place to the stomata. Now, this transpiration makes the water uptake or the water transport system. How? Water is lost from leaf pore in the form of transpiration. This decreases the concentration of the water, so the water from the neighboring cell moves in. Now, as the water from the neighboring cell moves in, that decreases the concentration of water in that area. So it takes water from the neighboring cell, and then from the neighboring cell, which makes a long continuous column, and eventually it forces the water from the soil to comes into the root, and then the xylem carries up due to transpiration stream. Okay, now what are the factors that affects transpiration? It is temperature, light, wind, and humidity. With increase in temperature, rate of transpiration increases because the particles gain kinetic energy, they start to evaporate faster. With light, rate of transpiration also increases because remember, as the photosynthesis will increase, the transpiration will increase, because as the rate of photosynthesis increase, stomata will increase to take in more carbon dioxide, which increases transpiration. Wind, as the wind speed increases, it moves all the saturated air, increasing the concentration gradient of the water vapor, increasing the rate of transpiration. Humidity, as the rate of transpiration decreases with humidity. Why? Because humidity will make the air saturated with water vapor, decreasing the concentration gradient, so the diffusion rate of water will slow down. Okay, now how do we measure the rate of photosynthesis or transpiration? I think you must have done this experiment. This is a potometer. A potometer, there's a glass tube, there's a reservoir of water, and there's a beaker, and there's an air bubble. As the water uptake, as the plant will lose water, it will try to uptake this water, and this bubble will move. You will use a scale and measure how much the bubble is moved. The movement of bubble is an indicator of water movement, which in turn shows the rate of transpiration. So we assume all the water that it is uptaking is equivalent to the rate of transpiration of water. Okay, now since transpiration is a necessary evil, why? Because transpiration is required for the uptake of water, but it can also cause the plant to unnecessarily lose the water and can wilt, they can droop. So the plant has done an adaptations to limit water loss. They are covered with the cuticle layer, which is waterproof and prevents the rate of transpiration. They have stomata on the lower surface that reduces the rate of transpiration. Some plant stomata opens at night and close during the day. In some plants like cacti, the leaves are reduced to spines that limits the rate of water loss. And some plants have sunken stomata, where the stomata is in pits, and they have the humid air around it that prevents the rate of transpiration. Okay, so I hope this unit is clear to you. Now it's the time to test yourself. In this unit, we have seen a lot of key terms. So you should be able to explain these key terms. So these key terms are cell, tissues, organs, organ system, digestive system, esophagus, stomach, liver, pancreas, small intestine, large intestine, substrate, active site, carbohydrates, protein, fats, enzyme, catalyst, denaturation, amylase, protease, lipase, bile, emulsification, plasma of the blood, red blood cell, white blood cells, platelets, plasma, arteries, veins, and capillaries, double circulatory system, coronary artery, coronary heart diseases, stent, statin, pacemaker, xylem, phloem, transpiration, translocation, potometer, stomata, and gut cell. So you can pause the video, have a go at these terms, and then check yourself if you know these terms. You can come back to my website. The link is mentioned in the description box below, and you can have the answers of them as well. Okay, so what is the next step? Next step is check the specification. Make sure whatever thing is there in the specification is crystal clear to you, and do exam questions on this topic. Okay, so I hope you have understood this video well. If you still have any doubts, leave a comment below. We'll reply you as soon as possible. Or you can go to our website where we have 24/7 chat support, where you can post any queries before your exam and you can get instant reply. You can go to my website where you can find the notes of this as well. The link is mentioned in the description box below. If you like this video, then choose subscribe to our channel and do not forget to like, comment and share this video. And if there's any specific topic you want me to make a video on, do leave a comment below and I'll make sure I'll put that up before your exam. Okay, so please go over this topic very, very seriously. It's a very important topic and I'll see you next in your next video. Till then, happy revising.